Welcome everybody. Today, we're going to talk about AV manufacturing and CMC in this Meet the Experts session. Um, this is a topic with, endless complexities and lots of questions around why this is so challenging and expensive. So, you know, Many of you know, I mean, I've been working on AVs for a number of years now, but like many of you, I'm learning about scaled manufacturing, CMC, and regulatory requirements as I'm going through this project with our Prion project and bringing it to first in human studies. So I've invited a couple experts from Viralgen, um, a, which is a CDMO located in San Sebastian, Spain, to this discussion. So Andy Holt, who we're waiting for, is the Chief Commercial Officer, and Jacob Smith, who's online, is the head of the technical development and CMC strategy at Viralgen. Many of you know Sonia and I are working with Viralgen to manufacture our AAVs for our Trailblazer Prion epigenetic editing program. And they've been really wonderful partners so far. This isn't meant to be any kind of sales pitch or anything. This is really meant to be an educational session, and I'm really hoping that they can share a lot of best practices around, working with CDMOs and just getting through AAV manufacturing and all the CMC and regulatory requirements with as few challenges or as few issues as possible. In terms of the agenda, Andy and Jacob are going to kick off the discussion with a primer on AV manufacturing. If Andy doesn't join soon and we don't have slides, we'll go straight into a discussion of technical things. And then, we'll also discuss the process of moving forward from, where we are in the lab with proof of concept testing of our AV gene therapies to working with the CDMO to manufacture the materials for initial assessment, toxicology studies, and for clinical use. And then we'll open it up for questions from everybody in the audience. So feel free to either add any questions you have to the chat, and Mindy, maybe you and Angela can help organize those for me, or better yet, just ask them live. Always rather just do that if possible. But if you have questions that you want to get answers to, and if you have to leave early or something, pop them in the chat. Oh, there's Andy.
Hey, sorry, y'all. I just had some technical issues getting people and the internet to behave, but they're sorted out now.
Sounds like a persistent problem. Great. So, Andy, just in time, I just gave a brief intro of what the agenda was going to look like. I think now I'll turn it over to both you and Jacob to give brief introductions, and then, I know you guys had prepared a few slides to serve as a primer for the discussion, and then we'll get into the discussion and then do a Q&A at the end. So I'll turn it over to you guys and let you introduce yourselves. Andy, do you want to go first, or Jacob maybe? Okay.
No, Jacob's smarter and more interesting than me, so I'll go first so it doesn't intimidate. My name's Andy Holt. I'm the Chief Commercial Officer for Viralgen. Background, I'm a bad molecular biologist that got onto the commercial side of making cell and gene therapies about 17 years ago. Been through a number of CDMOs, starting from kind of smaller groups in the early days, making a bunch of adenovirus and lentivirus, and then some envelope viruses and vaccines. And then as the field has evolved, I've spent more and more time working with AAV to the point that in 2017, I joined AskBio, helped create Viralgen as an AAV-only CDMO, and have been working ever since to help try to promote better access and make manufacturing a little bit more boring than it can be. So I'm super excited for the opportunity. Thank you guys so much for the chance, Ben, Sonia, and the whole team. Nice to see some familiar faces on here, and hopefully this is useful.
Hey everyone, Jacob Smith, nice to meet you as well. Happy to be here and talking with the group. I've had the good fortune of being most of my career in AAV process and analytics development, mostly on the product and therapeutic development side. I started out working in AAV mediated gene therapy at UNC Chapel Hill, and I worked through the groups there at the Vector Core and Gene Therapy Center before jumping into the private sector where I was on process development teams with Bamboo and Pfizer and really working on a lot of the earlier iterations of scalable AAV based or scalable 293 based transient transfection production platforms. And I think in the last 15 years, we've seen like a lot of everyone chasing larger scales and newer technologies. And I think the exciting thing in the industry is that we're moving past some of the manufacturing bottlenecks, and it's how do we take the processes that exist in the industry and then improve the accessibility for a lot of the different therapies that are out there. And importantly, what can we do with the data? So I'm really looking forward to getting into some of the questions that Ben has set up for us today, and hoping to share some of our experience and insights and help advance programs along the way. And with that, Ben, I'll turn it back over to you.
Great. Well, I'll probably turn it back over to you guys. Do we want to start with the slides that you guys have prepared for the primer?
Yeah, I can bring those up, Ben, if that works. And our intent here is just to give you a little bit of background about who we are, and then some basic pieces of like working through this process of scale up and the decisions. We've been really, really lucky to work with a number of groups that are like kind of passionately committed to rare disease, and we found that those groups have a very broad set of skills, and they're not always CMC people, right? Sometimes I'm jealous of folks who aren't CMC people because you don't have to deal with this stuff all the time. And then sometimes, not to call it out, but Steve Gray's in the corner of my screen. Sometimes you deal with like people who have really deep, decades of CMC experience, but it's still the same. Hopefully part of what we're here to be helpful with is just to say what to look out for, what can we do different, and then also to listen to y'all about what worries you and what have you seen, and maybe how we can do better as an industry to help support projects. Because I think without getting too editorial, the world's a different place than it was a few years ago. Projects are moving forward into the clinic meaningfully from many different places that it didn't used to move. And it's up to us and groups like ours to make that easier and better, to lower those barriers to entry. So hopefully that's what we accomplished today a little bit. So I'll bring up my screen here and see if this is useful. Let's see. So let me know if this is coming up okay and it works all right, and you're seeing some pastel Miami colors here in our current branding, and
or at least I see it. Yep.
Perfect. If we've broken the internet, Ben, if you can see it and nobody else can, so that's great. So Viralgen started out as a group that was a joint venture. AskBio spun it out to try to solve some of their financing and manufacturing problems simultaneously. That joint venture was consolidated, and we've been around as a profitable and growing entity based on the north coast of Spain. San Sebastian is not a biotech hub. You haven't missed that, but we're helping to try to build one there. The reason we ended up there is really because of rare disease, because of San Filippo, as well as to try to help build a biotech community in the Basque region, which was honestly pretty heavily incentivized for us when we went there. But it's a special place. We've got now close to 500 employees working on gene therapy, on AAV together, and trying to commercialize our first drug here later this year. Our vision for this was to do one thing and to do it well. Fundamentally, again, without being too much of an editorial comment, I think that the basic CDMO model doesn't work for advanced therapies. The drugs are too complex. The processes are sometimes underdeveloped, and a lot of times you get positive signal from a molecule way earlier than you expected, right? The catastrophic success problem is something that's plagued the industry for a long time. So if we could do one thing and simplify it by really building a platform and then running it to allow for context, to allow for knowledge to be gained, and to allow for a CDMO to own what they should own, which is making the drug. So our vision from the start with this platform, we call her Ava, is you hand us a plasmid and we hand you back a product and we handle the rest. Others are doing this. The field is maturing, and it's really exciting to see this idea propagating across the industry, propagating into the FDA with platform designations and other kind of bells and whistles. But this is the vision, that you should be able to hand us fundamentally your gene of interest, and we hand you back a drug. And I think if you're looking at CDMOs, this idea, this concept of the manufacturing process as the CDMO's problem is not universal, but it's something you need to consider as one of two possible options, right? Either you're handing your process to a CDMO, or the CDMO has a process for you to use, and there's pluses and minuses to both. But this is certainly our approach. So you're going to hear an inherent bias from both Jacob and myself towards this approach, because it's one that we've tried really hard to build. Our process starts with a good cell line, but it's a triple transfection process. And these unit operations, I think, are things you've seen throughout the industry. When I say trying to make manufacturing boring, it doesn't mean we're not implementing technologies, and Jacob doesn't spend a ton of his time thinking about technology development and improvement. It just means that we need to be very diligent scientists and only change one variable at a time, so we don't leave our customers hanging. We don't create a situation where we don't know what is going on in our system. So almost everything we do is kind of off-the-shelf pieces optimized for AAV or optimized for a serotype. And Ben and Sonia, the team, have been through a ton of this with Viralgen as we've worked to optimize their capsid. For them, it was an upstream step that was a key unlock. For other serotypes, it's been a downstream step that's been a key unlock. And that's kind of the process of evaluation and optimization, not really development, is a key thing to consider. That if you're doing something that's a little bit outside of the norm, right? If it's not a AV2689, that a CDMO partner may need a little bit of extra time to play with it and to get familiar with it, whether they have a platform or not. Jacob, do you have anything, since you're one of the guys who invented this process? Anything else you want to say?
No, I mean, I think you covered it well. I think the only thing I touched on it really briefly in my introduction is that, I think when this was developed, this was created at the time where everybody was trying to get a scalable manufacturing process. I think circa 2012, 2013, like all AAV demand was massively on the rise. You couldn't have enough manufacturing capacity. And so I think, there was this huge demand for the technology to really leap forward, get out of like the adherent systems, roller bottles, even some of the like more modernized versions of those things like cell stacks and iCELLis reactors and all of that. This was really, how do we have a unit operation flow that looks more like a standard biologics process, with a couple of exceptions. So obviously things like the iodixonol density gradients, they're still serving a purpose. And I think if you ask me in 2014 where those will be in 2025, 2026, like everybody that was in the space at the time would have said, now they'll be completely gone. And now I think we have a completely different perspective on it. And I think that highlights like what Andy was saying too, around like, how do we really look at what is meaningful change when it comes to like AAV production and analytical technologies, and like what today mobilizes the field forward, mobilizes programs, and improves accessibility. And it's a little bit different. Like back 10, 15 years ago, it was we need something scalable. We need something that produces more than anybody else is doing in the world. And now I think some of those science questions and science challenges have changed a little bit. But like Andy said, it's not to say that we're not still iterating around how do we make the best products. It's just not always chasing productivity anymore.
Yeah, I think the key to a lot of this for us and for the manufacturing industry generally is that we're trying aggressively to move beyond product specific process development as manufacturing companies. Having a bespoke process for your vector, right? And adding all those variables and trying to kind of play whack-a-mole with if you're seeing manufacturing variability, is it your analytics? Is it your vector? Is it your process? The more of those things you can hold constant, even if you may be sacrificing the platonic ideal for yield. Um, we've had this conversation with Ben, we've had this conversation with almost every one of our clients, right? We could spend 6 months and improve yield, or you could be in the clinic. And those are really hard discussions to have, especially with innovators, right? With folks whose life's work in some ways is devoted to this disease, to this technology, to this whatever. Those are challenging discussions, but I think it's something really worth considering up front about where your returns start to diminish and where you can reach those kind of inflection points. If this was a group of biotech investors, I would say value inflection points, but for this group instead, I would say kind of development inflection points where you get that first in human data, where you get to learn the limits of your drug in the only system that you can learn those limits. And I think so for us, our burden that we choose to take on in a lot of these situations is to help find those points of diminishing returns and spend the appropriate amount of time on the clarification step or on the affinity step or considering moving to our higher yield platform, right? Which is perfusion based and doesn't have an iodexinol gradient and these systems exist. I'm not saying anything that other companies don't have too. This is not totally unique, but there are higher yielding but more variable systems out there than our base platform, than the platforms that other companies in our space offer. But you have to find that balance, especially when I think for all of us, we're in a race against time for our patients, and we're against money, to be blunt, about funding and those sort of reaching the milestones that help enable the next stage of your development. That push-pull is something that needs to be considered, and we spend a lot of time talking through. So, this is just a little bit of a why should you believe us? You don't have to. It's still optional, but I would say, we've run this platform close to 2,000 times now. We're in 44 clinical trials around the world. This approach has helped. I'm proud of what we've been able to do in this space, especially in the last few years in a challenging environment to help drugs get to patients. And a lot of it has been because we've stuck to this idea of platform. As I said, there are plenty of people who don't work with us, and I'd be happy to introduce anybody in the world to 3 or 4 really good CDMOs out there and make sure you get exactly what you need. But this expertise we built in AAV, we've come by it honestly, and I'm proud that we've been able to get to some really interesting milestones as a result for the folks we work with, both in academia and nonprofit, as well as for big biotech and large pharma. So we've made 30 different serotypes, thousands of batches. We've learned a lot about our platform as we went and made it better every time, which is also the exchange every one of our customers has to be okay with, is the next batch is always going to be our best batch, right? Because we get to roll everything we learn into the next one. Down in the weeds a little bit, and this is a very wordy slide, and I can give you this slide later so you don't have to worry about it. But if you start to work with a CDMO, you should look at it in kind of 3 phases, right? And the first phase is really kicking the tires. This is kind of the demo and R&D batch. For us, it's a 2-liter. For a lot of places, it's something between a 2-liter and a 10-liter. And this should be cheap. This should be fast. This should let you see your drug, your therapeutic in the hands of the CDMO for the first time in a representative platform. It also lets us as a CDMO evaluate you. And this is where, in this stage, you're visiting the CDMOs. Please go visit. Please prioritize the time to spend time on site wherever you're making stuff with the team, getting to know them, meet your project managers, meet your head of quality, meet your head of manufacturing, look those people in the eye and really make sure you can start to establish a trusting relationship at that stage. And then this is also where we can kind of have the discussion back and forth. I have what to worry about and get excited about here because there's always things that stress you out and things that are really cool to see. I think starting to think about plasmid quality at this point, if you're going to be in a transfection-based system, is key. Starting to think about how you're going to negotiate the contracts. If you're in an academic setting or a grant funding setting, you're going to have bureaucracy to navigate to transfer hundreds of thousands or millions of dollars back and forth from your entity to a different entity. And your CDMO is going to be handling your intellectual property. So somebody in your licensing office is going to be annoyed about that. You're going to have multiple stakeholders that start to enter the party at this stage. And if you want to move fast, you need to start thinking about your master service agreement, your quality agreement, who's going to review it, who's going to sign it, all of those fun things on the business side. You also need to start thinking about logistics, right? How are we going to move biologically hazardous materials at a frozen temperature around the world? Your CDMO should be able to help, but you should start thinking about, what freezers is going to go in, what are we going to do with it, all that sort of stuff at this stage. I also want to say that likely this is the last stage. You have to make a lot of your CDMO confidence decisions at this stage. So take your time here, make sure you feel good about it, because from then the train does kind of leave the station. Some CDMOs call the next stage toxicology, some are engineering. It's all the same stuff. It's a batch used in those animal studies that help prove the safety of your drug. A good CDMO should ask you to do a lot of other work with that material on the analytical development side, on the stability side. There's a lot to think about there on especially potency in your transgene ddPCR. And, the what to worry about, you really have to start worrying about pre-IND stuff, right? Your tox study scope can impact your timelines, impact your cost. All that stuff has to be ready before you really finish the tox batch for sure. How you do plasmid, like what grade of plasmid again comes up, that's a theme throughout. How long you do stability on your tox batch is a key piece, both in terms of material consumption and in terms of clinical readiness. And then, some of the more practical stuff, right? Are you going to send somebody over to be a person in plant to watch the process? Are you going to be actively involved in supervising the manufacturing? Some of our clients have gotten FDA feedback about, control of CDMOs, and you need to demonstrate that either through a person in plant strategy or through observation. There's a lot of ways to do it, but you need to think about whether you're going to go or how you're going to do that. And if it's you or if it's a consultant, really think about the GMP batch, because the train has fully left the station at this point. And then for GMP, a lot of these decisions get made earlier with people like Jacob deeply involved, but if you scale the manufacturing right for many of these indications and applications, you could supply your whole trial off of a single batch. You could at least supply multiple phases of an accelerated trial design off of a single batch. You also have to think about all of the supportive studies that come in, right? All of the clinical stuff, all of the device compatibility. There's a giant list of stuff we could get down in the weeds on here, but there's a ton of other supportive studies that come out of the GMP material that you need to consider. You also need to have somebody ready for quality, because batch disposition is going to be an ocean of paperwork. All of that paperwork is important. The FDA will see almost all of that paperwork. So who do you have to support you there, whether it's on the CDMO side or whether it's independent or both? Really be thinking about that as you get close to the GMP phase. I'll pause there, Jacob. You've done a ton of these. Any other pain points that I've missed here? Any things to think about?
No, I was trying to think of specific examples as you went through. I'm glad you called out device compatibility. As a part of the supporting study piece. I think like the pharmacy manual and the device compatibility, like at least in my experience on the CDMO side, and then even on like the therapeutic developer side that works with the CDMO, is that was always something that was kicking back and forth as to like who really owns that, because that's after the material's been released and shipped to your clinical storage depot or wherever it's going to go to prior to the pharmacy. And so, but knowing that it's compatible with whatever the delivery device is a critical study, and then obviously all the analytics that you would do to show that it's acceptable are CMC methods. Um, I think the one overarching theme that I would put on this slide specifically is um, those earlier boxes. There's going to be a lot of changes there. The one thing I think that's been tolerated well in regulatory filings and in interactions with the various like health authorities is that like CMC changes are okay. The thing that I like to encourage the therapeutic developer to really think about is like what amount of material do I need at each of these pivotal steps throughout this life cycle, and am I getting the right material for the different studies? That like I think if you have a good manufacturing partner, they're going to alleviate like the how you get all of that stuff. But I think having clarity on like the scope and the overall end-to-end plan is what's going to be the most meaningful because you're going to have some changes. Like some changes are normal and they're tolerated. But just trying to anticipate where those are at and then early adoption of that sort of GMP mindset of like, okay, well, if I'm going to tinker with the construct really up here in R&D, early R&D work and construct development work, that's fine. But if that material then, if it was made in-house or if it was made at your CDMO, where did it come from? What studies did it go to? In some capacity, you might have to bridge that. So I think I would say like don't make a bunch of pivotal dose range finding decisions off of material that's not representative to what your future clinical lot or your toxicological study lot is going to be made at. It's consistency is key through here. I think planning, clarity of the scope, and consistency.
Thank you, Jacob. I think there should be a lot of kind of knowledge to pull from in a good partner across this scope, right? And that's where, for us, it's always interesting because we see, eye and ear customers basically behave the same, have a lot of the same problems. Almost everybody dosing into the CNS has at least 75% of the same problems and challenges and questions. Almost everybody doing systemic for neuromuscular looks very similar from a CMC perspective. Everybody doing systemic on metabolic looks pretty similar from a CMC perspective. So there's always lessons to be learned and kind of knowledge to be leveraged. Because ultimately the mechanism of action of the drugs tends to be pretty similar. Everybody doing gene editing has the same challenges, right? Or not the same, but like many of the same challenges. The manufacturing process doesn't care why you're oversized. It just knows you're oversized. So let's work together on fixing oversized problems. I think every CDMO can be challenged to say, well, if all you have is a hammer, all you see is nails. I get that, but for us, we really do, the rise of modality specific CDMOs in cell and gene over the last 10, 12 years, I really do think fundamentally offers better options than it used to across the board and uniquely better options, especially in viral production. Because we all are getting a lot more experience and we've seen it, but all of our competitors have too. Like the barriers are lowering, right? We're not as tight with information as we used to be. We're trying to be better. Speaking across CDMO. So yeah, there should be some acceleration to be offered by leaning on folks. And I know I'll speak for Jacob and certainly for myself, like here and throughout, if we can ever be useful, I don't care whether you work with us or not. If we can be useful, give us a call. Our emails will be available. We love working on these sort of projects. If you go somewhere else, you're going to hurt our feelings, of course, right? We'll cry, but we'll cry privately, and then we'll get to work for your stuff because we love seeing these drugs move forward. So if we or referrals or anything we could ever do, we'd be happy to. Last slide before discussion, I just wanted to present kind of what to expect in terms of timelines and where you have some exit points. And this is a real drug but I had to strip everything off for confidentiality purposes. But this is a weird capsid. It's nobody on the call's capsid. It's somebody else's. But we did need to do some design of experiment and optimization work. You should expect the period of kind of first touch to, those demo or small scale batches being completed being 2 or 3 months. Just because I think Jacob put it well, like something always changes, something's always going on. There's a shipping or logistics thing. We all learn about each other during this phase. So 2 or 3 months to kind of get your first hands on material as CDMO makes is reasonable. You'll see here that the reality of the rest of CMC is we spend 20% of the time manufacturing and 80% of the time characterizing. And there are a couple of real pain in the butt assays that take up almost all of this time. So there's data pouring out of the batch throughout that characterization period, but 4 to 6 months to characterize a batch is good. Like closer to 4 is better, of course. We've done it in that routinely, but there are some assays that take 45 to 50 days on test plus reporting. Those things take time. So especially when we're pulling samples right throughout the manufacturing process, and there's a number of tests that are done on the final product, so we don't have samples available until the last day of manufacturing, literally. And then we have 50 days of testing. That really adds up. So just know that throughout this process, you're going to have data and I don't know, Jacob, when do you say you breathe kind of the sigh of relief? I tend to breathe one when it's in the vial and one about a month later. Then everything else from there gets pretty boring.
Yeah, I think that big first one is it's in the vial. And maybe in the vial, and I know like the bulk titer and the DP titer. And then a month after that, or 3 weeks after that, you have probably 80% of the methods. Like there's a couple of the like just inherently longer range methods that take a little bit longer. Sterility is one of them. But that's one where we are never expecting a failure there. Like, so it's kind of boring. And so if we get past the purity and the strength and if you have potency as a part of strength, then that's when I start to breathe a little bit easier. Um, yeah.
Makes sense. The only other last piece I'd say is I think CDMOs generally have gotten a lot better. When I was in a couple of previous lives ago, we had a commercial drug that in cell therapy that was 60% of the batches failed. Good CDMOs now are releasing 80 or 90% or more of their clinical batches that they manufacture. Our batch failure rate is less than 5%. Good CDMOs in gene therapy should be able to deliver. So when we say the blood pressure goes down here, it's just because, every now and then it's good to take a step back and realize we're tricking cells into making virus that doesn't want to be made, and that's hard. So there's always a deep breath coming when the batch finishes. I have these 3 stars here just to highlight too that if you want to make a no-go decision, this one here is the important one, right? How much time you spend in this blue phase, and then this decision is a key one that the more criteria you set back here for success, the easier this decision gets. But deciding to stop here is very hard and expensive. You can do it, but it's hard and expensive. The worst case, of course, and we inherited a project last year where a customer unfortunately had a failure here, and then we had to jump in. Doesn't happen often, but it does happen. And when that happens and somebody's switching CDMOs, these timelines the Gantt chart kind of starts to look like spaghetti. It doesn't look good anymore, but you're able to get it done. But just to also emphasize that I think this expectation for timelines of kind of first touch to GMP ready in a year is reasonable, I promise. There's always things that could make it go a little faster. There's things that could make it go slower. If you're doing your tox study in its entirety before you start GMP manufacturing, as an example, then you're going to be paused here until your tox study is complete. That's fine. Many customers choose to do that. But just to emphasize, I think these timelines are achievable routinely in the industry. So long as you have your plasmid act together and your analytical development act together, you will be, everybody can do this. So with that, Ben, I'll probably stop because I just have discussion points and we're on to your phase of the discussion. Hopefully that was worthwhile.
Yeah, that was great. Thank you, Jacob and Andy. Maybe while we're on, I was going to go in a slightly different order. While we are on the Gantt chart, and if you want, bring it back up. If not, it's not required. One of the questions that came in prior to this from the SCGE group was, for phase 1 drug products, there are minimalist approaches to reduce costs and better enable first-in-human studies. However, this can lead to CMC delays at later stages of product development, and commercialization. Are there any low-cost, high-impact things that can be done early to accelerate the future product development and approval, assuming phase 1 trials are successful? So where all of us here are working obviously on a NIH-funded project that is very generously funded, but we do have limited resources. Where can we save? Where are the possible money saving that won't shoot us in the foot down the road if we want to commercialize?
Andy, I mean, I think I can give maybe from the technical perspective, but then I think obviously it'd be good for you to weigh in on this just because you see what everything costs and they don't typically let me talk about that. So, and I prefer not to. But I think like Andy highlighted this somewhere in the slides earlier is like early on take the minimalist approach. Like I think the real watch out for like your low cost high impact decisions there are having I think having the discipline to not over customize, especially when it comes to CMC. Focus on the essential CMC activities that are in phase 1 that were in that previous slide that Andy hit a lot of those earlier and it's kind of listed out. And then accept the fact that like maybe there's a comparability component here because maybe what we really need to do if it's a large indication that does require a little bit of process development or process optimization. Maybe you want to move from like our base case process to a more advanced production system just because that it's going to be too expensive. The therapeutic itself is going to be too expensive because we need so much of it. And that manufacturing process it's not delivering what you need. That's okay. I think that's certainly something too that if you have a manufacturing partner that has different levers that they can pull or has different offerings when it comes to process, ideally that's a framework that they've already mapped out. And in the case of Viralgen, and I'll try to not present this as like a pitch for Viralgen because I think many of the other CDMOs that are moving in the direction of platform technologies are doing the very thing of where let's use our flagship process. This gets you quickly into the clinic, and gives you the early readout on proof of concept and safety, gives you a little bit of visibility to dose, and then really start locking in on process and later on. So I think that was a long-winded answer, but I think like the biggest thing there is avoiding the risk of over customizing for your specific product. Like if you can really look at it as like, can I launch this trial with the base process, minimal efforts going forward, only focusing on critical CMC activities, and then maybe customize a little bit later, but based off of need.
Yeah, I think the only thing I'd offer too, I mean, investing in kind of an analytical strategy and stability strategy will help make you future-proof. I think, and that's relatively inexpensive because you can always validate so long as you create the right foundation, you can always build upon it, right? So if you're like thinking about titer and potency, you don't have to have fully validated assays and nominal titer and all that stuff right out of the gate, right? But you will have to rework those assays if you didn't build them right in the first place, anticipating titer going to nominal titer, anticipating a cell-based potency assay needing to be validated. I think some thoughts on that stuff early will put you in a good position with the regulators and allow you to accelerate, because that's really what I'm always looking for is, like I mentioned it before, like the catastrophic success problem, right? Where that fourth or fifth patient, right? So you're in dose escalation, you're seeing a linear response to your therapeutic going up in dose and a therapeutic effect increasing in magnitude when your dose goes up, and you're still under the safety limit. So you have like those moments are magic, right? That you get all green lights across the board, and then oceans of possibilities are now open to you. And in previous generations of gene therapy development, you're then saying, well, I ran 45 hyperstacks, and 3 of the guys in the vector core have rotator cuff injuries, so they can't do hyperstacks anymore. And I can't find serum because my New Zealand guy went out of business. And oh my God, what do I do? And that's a real story, as a side note. That's, that happens. It still happens. But the world is a different place now, and I think you can future-proof a little bit better for the regulatory agility that's open, and the potential that we understand these drugs and the process better than we did. So I'd call focus on don't be dumb about raw materials, invest smartly in analytical development, and think carefully about what happens if you're right.
Great. That was super helpful. I guess I'll take us back to the very beginning of the process just for a second. I'm sure there's people in the SEG consortium that are at all stages. What advice do you guys have for academic groups that are during the CDMO interviewing process? Like, I remember when we were interviewing lots of different CDMOs, it was comparing apples to oranges to bananas to every kind of fruit, right? The way that the pricing structure worked, the offerings, the way they measured yields, everything just is so different between all the different groups. It makes it really hard for those of us who haven't gone through this before to make the right decision. And so just curious if you can talk about that process from your perspective, and also like what are some of the red flags we should look out for, in terms of hidden costs and things. I mean, I know you guys, your platform is meant to be rather inclusive, and so there's a benefit of that, and you said that a lot of CDMOs are moving in that direction, but we certainly talked to other groups that were much more a la carte in terms of pricing and didn't necessarily include a lot of third party pass-through costs and things like that. So maybe if you could just talk a little bit about what we should be looking out for.
Yeah, I mean, I think just on the pricing and kind of model side, to say the quiet part out loud, that's probably on purpose, right? That a lot of these things are obfuscated to the point that a direct comparison is challenging. That doesn't, if I'm being charitable, I would say that we're not commoditized yet, right? As much as I would like to be, where in other modalities, right, you could say, okay, I need 4.8 grams per liter and I need a 2K, and I want to see a stable cell line, and I want to see these, what's the pH condition of your protein A column? Other modalities are more commoditized, are more competitive on that, where it is, it's both easier for the vendor and the customer to compare apples to apples. For us, even a few years ago, multiple production systems, multiple IP landscapes, multiple bioreactor manufacturers, right? Like, I don't know how I think Jacob and Steve, maybe, I can't see everybody on the call, but like, there may only be 10% of the people on this call who have a strong opinion about Sartorius versus Thermo bioreactors, and half of your opinion is arbitrary because you had a bad experience 6 years ago with one or the other and you never went back. All of that informs the apples to oranges problem. So I think a lot of this is look at data and performance, look at employee turnover rates, look at batch, on time and full delivery rates for the entire business. Look at specific experience with your serotype and the serotypes next door to it, right? Almost if you're dealing with a novel serotype, I guarantee you it has, 85% homology to X, Y, or Z. Ask for those data sets too. Um, all of that will help. Um, the other piece is in terms of cost, that's almost to me more of an opinion question. Like you're absolutely right, Ben. If we work together, there's no way for you to tell when you're working with us, how much our helper plasmid costs because we provide it. It's included. We think that delivers value to you because if we use X or we use 2X, that's not your problem. But it's entirely opinion based whether you think that's valuable or not. And everybody on this call, everybody in the world is entitled to their opinion, right? If you want to control how many hours your project manager spends on the project, there are CDMOs out there that line item everything out and you can control that. If you want one price and it's probably a little more, half of the people we talk to feel that's opaque, half feel it's transparent to say, this is the money, this is the product, we will deal with it. I would say our model requires more trust for sure. And we've struggled with that at times. We've definitely screwed up conversations with customers where it didn't feel like transparency to them. It felt like we were trying to get one over on them, right? And they're entitled to that. I will just say our model and the platform model generally is meant to simplify, not necessarily make more transparent, because I don't know why you would care how much helper plasmid we're using. But every model out there is valid. So just think about what feels right to you. And I wish I could make it be more like data driven or more of a head decision. But for me, that model question is really more of like a heart and a gut decision of if you're sitting across the table from somebody and they say, this is all included. If you believe them, or you go talk to a couple of their other references and they say, yeah, no, it was great. They delivered. There was no follow on. They didn't nickel and dime us to death. It worked. Then figure out how you feel about that. And if the references don't check out or you have an experience where you do feel like you're getting in a taxi cab, right? And the longer you stay in the cab, the more the meter's running. And you don't have the bandwidth to manage that and manage those costs on a granular level. This is, like I said, it's much more of like a heart and gut discussion than it is a head discussion. I think evaluating the technical expertise and the ability to deliver of the CDMO is a head discussion. And I could give you, or Jacob could give you 100 questions to go ask and make sure that you're getting a good one. There's a bunch of good ones out there right now, honestly. So, but the rest of it, the model and the people and all that stuff is heart and gut stuff. So I don't know, Jacob, if you feel different.
No, I think I had a similar answer. I mean, I've gone through this. I've been on the other side of that as well, and done what Andy has highlighted, where I mean, you literally have a table of evaluation criteria that are based off of process and starting materials and analytics and quality management systems and GMP, and timeline and cost is on there, because like first and foremost, you need them to hit your timeline. If that can be a non-starter. So having an understanding of generally what the timeline looks like is good. But then, yeah, I agree with Andy that, cost is going to be different across all of the different ones. And, I would say like historically, like if someone was very vague about cost, like that could be perceived as a red flag. But that's where I would say like really make sure you've scrutinized all of those other areas really well first, and you understand that if I'm getting something that is of high quality and it meets my timeline, and I have a good feel about it as far as where the cost is and in the context of my budget, then like I think you're in a good spot there. I think the only other like specific thing, like without going too detailed, that I didn't hear Andy mention because Andy gave a really nice overview, and he's worked in CDMOs longer, so he knows he knows more what to ask than I do actually. Is facility. I think looking at capacity, I think like Andy did scratch on this a little bit with like headcount and personnel attrition. But like having an understanding of like what their capacity to deliver your product at all phases in development and be, and into commercialization, what that looks like. And then what the current iteration of the facility and what's the future iteration of the facility and how that changes. Um, and then I think the last piece that I would mention too, that I don't know if you mentioned this, Andy, is like, what does the tech transfer situation look like? So, are they open to, accepting your process if you really want to go down that route? I think we've hopefully sufficiently discouraged over customization of process. But if you still want to go down that route, then like, how do they handle it, and will they work with you on it? Do they work with other CDMOs? If you partner your program with a large pharma partner, will they transfer to that for internal manufacturing? So I think having a good understanding of how tech transfer from a manufacturing standpoint is handled is also a good thing to go into the discussions early on understanding.
Yeah, great. Maybe I'll dig a little bit deeper on that last point. I mean, many of us are working on, we have funding through this program to get our programs to an IND. We don't have funding for the next steps through this program, right? So, I think if our first in human studies are successful, there's potential for these to be, spun up as a startup or partnered with some other pharma or biotech company. What disadvantages are there? Some of them I guess are kind of obvious, but what are the advantages and disadvantages of having a platform process that is somewhat proprietary with one CDMO, and how does that influence, the later commercialization or picking up and having a partnership to advance that therapy? If they may want, a company may want to come in and use their own processors, their own CDMO.
Yeah, I mean, I think ultimately a lot of that becomes almost the CDMO's problem to defend, right? It's Steve. Sorry, I didn't see you had a hand up, man. You got.
Yeah, Steve, go ahead.
Well, no, I was just going to add on to that of, it's always when everything is going great, it's going great. And then when it's not, then it's really not. And, so it's like what happens when things go sideways or something. And, I guess with this idea of like a tech transfer out later, how do you, I guess, if you don't bring that up initially, how much are you really killing yourself or introducing potential risk to kind of, because I can see if you try to pre-negotiate that on the outset, it can get really complicated, but without pre-negotiating on the outset, then you're really at the mercy of the CDMO in terms of how they'll handle that later on. So yeah, any advice there?
Yeah, I mean, if you want my like unvarnished answer to that is it doesn't matter who owns the platform. Transferring out of a CDMO sucks and is painful and is going to be a giant pain in the ass no matter, like, congrats, you own your platform. It's still going to be terrible. Because in real life, right, the only times you're transferring out are like both catastrophic and repeated failure.
Mhm.
At least in my experience, right? The only times we've inherited projects from other CDMOs like midstream, or we have lost projects in CDMO to other competitors midstream is when we were just god awful, right? There is an enormous inertia. And that's why I think making the CDMO selection process is so important and why it's probably so stressful for everybody here. Doesn't matter who owns it. Really doesn't. Moving it around is going to be terrible. So my bottom line is that the evolution of comparability standards and flexibility in the last few years should be a huge source of encouragement and optimism for anybody looking at the CDMO industry, because the better your analytics are and the more consistent analytics are across CDMOs, right? We're all starting to finally agree that like AUC is a good protocol and that transgene ddPCRs are necessary and that a cell-based potency assay is probably the right idea, but not with some some weird cell line that doesn't work. These consolidations, right? This convergent evolution across the industry, whether it's bird wings or bat wings, everybody's trying to fly. So I think that from a therapeutic developer side is, and like, again, I'll be transparent. If you look around at like our big competitors and everybody developing platforms, they're all doing 293. They all look like us. They all, it's all remarkably similar process steps and remarkably similar approaches to this stuff, which is, for me as like a human, I love because it means more drugs are getting to more people. For me as a competitor, it's harder to find clear air around us that makes us uniquely qualified. So your ability to be portable, whether or not the CDMO owns the platform or not, is rapidly improving, so long as you spend the money on analytics up front.
Well,
So do that.
yeah, and can I follow up on that, because this is one of these things, if you're thinking about it, it can kind of create a lot of angst, right? Of you making sure you make that decision with a CDMO, because you're basically stuck with them for life, or it's going to be a real big pain not to be. But touching on what you said about some of the analytical comparability is, I think it seems to me like it's a lot easier to change manufacturers and change processes, maybe even going from phase 1 to phase 2, or from tox to clinical, or whatever. It's a lot easier to change your process and change your manufacturers now, because there's getting more analytical harmony out there to say that the products are comparable. But the important thing is that you maintain a lot of retains. And that's a big, big contention point when you're trying to have enough doses for your trial and you're fighting with the manufacturer over how many retains you keep. So I don't know if you want to comment on that a little.
No, I mean, it's something we struggle with too, right? Because we know every vial of material that we say, hey, put this aside for a day that you'll need it, is a dose that maybe isn't going into a patient to prove your point that you have a valid therapy. Like that push-pull, hopefully gets less stressful as yields continue to rise, as we get better and better process controls and better predictability. I think the problem will get easier as time moves forward. I think this is also one of those areas that in your pre-IND setting is a great thing to push on with the FDA, especially because we've seen flexibility around retain strategies there, or a retain vial could serve multiple purposes, check multiple boxes. There are some really good, aggressive, but not risky strategies to conserve retain materials and then apply them later. And you have to be really thoughtful, and this is way down in the weeds, right? But like, is a test a destructive test or not? Those sort of kind of tertiary analyses of your QCs can unlock a lot of vials if you want to look at them and spend a lot of time doing really boring work around destructive versus non-destructive tests. I think, to loop briefly back, Ben, to your original question, though, like, if you're going into late phase, you're going to need to really do a different evaluation of your CDMO. This is where I talk about future proofing. Like, if a CDMO during your initial visit can't talk about their plan for commercialization, it should go in the negative but not disqualifying column, probably, in my opinion. But you'd better have a plan. And everybody's going to need more than one commercial supply site. I know it's weird to hear talk about preclinical studies and NIH-funded studies and think about commercial strategies, but like, if you're going to ask me to wear like my most commercial hat I've worn on this call, I would say you're going to be a better partnership target and a better out-licensing target if you have that strategy baked in now.
That's great. Yeah, maybe I'll dive into a few technical questions before we just really open it up for everybody's questions. You know, many of us here are working on gene editing based therapeutics, and these come with their own set of challenges, often related to the overstuffed AAVs or larger than wild type AAVs. Um, what do you recommend for groups who are in that space trying to squeeze, a lot into a single AAV? Are maybe this is irrelevant now because, as you mentioned, a lot of the best CDMOs are kind of converging on very similar processes. But is it, these can be tough to produce, and I'm wondering if you advise groups to actually do small scale feasibility studies with multiple CDMOs. That could potentially get pretty costly, but what are the alternatives? What data could we be asking for from the CDMOs to have confidence that they're going to be able to create or manufacture a overstuffed or 5 kb difficult genome?
I mean, yeah, Jacob, if you got something to say, go ahead. I would say I recently asked our team to analyze if we never charged for small scale studies again.
Interesting.
Because I wanted to see the thought exercise. We probably can't forever or for everybody. But without making eye contact, we have been very easy negotiators with multiple people on this call to just do the right thing and deal with the consequences. So I would say, we do not make, and I would be shocked if any of my competitors make a ton of money on those small scale studies. It would be a crisis for our business if we did not convert most of those small scale study customers to large scale customers. My job is to keep our suites full. CDMO economics only work with full GMP capacity. So I will do almost anything to keep GMP capacity full. I would be shocked if all of my competitors didn't feel the same way because they have the same amount of dollars in economics that I do. And I may not be like the best at anything, but I'm pretty good at CDMO economics because I've devoted my life to it. Please read the context for what I'm saying in that statement appropriately.
Thank you. I appreciate that, Andy. That's very helpful. And certainly that's been our experience working with you guys. Maybe on kind of a related topic, I think one thing that I know some groups in this consortium and others working on gene editors are constantly struggling with is, do we put in the time to develop a single large vector, or do we go forward faster towards humans with a dual vector system? And this is a complicated question from scientific point of view, from dosing point of view. I'm wondering if you and Jacob can just talk a little bit about, from the manufacturing side, what are the things people should be thinking about if they're, to try to condense onto one genome, but with the possibility that may be an overstuffed, more difficult to manufacture AV, which is just a faster, easier approach. What should we be thinking about from manufacturing?
Yeah, I mean, I think so. I think if I broke it down like in the sort of like order of like preferred activities to like approach an oversized product, the first one, and this is an unfair one, but the first one is to right size it first. But I think if like given the question, I think we can't do that one. If we, I think that's where you do get into the decision or at least the evaluation of like you suggested, Ben. Do you kind of take the production hit, and then probably beef up the analytical panel beyond like maybe the standard panel to really understand like genome integrity and product quality. So for example, like that might be like multiplexing your PCR and mapping across the genome. That might also include NGS, which is great, but it comes with a lot of headaches because there's a lot of interpolation of what the data mean, and how do all of those things translate into actual like product strength and potency? So I think this is just my opinion, but I think, I think if you're going to go down the route of like considering producing either with a dual vector system, whether that's like at the genome level where you're splicing and delivering two vectors, or it's at the protein level of reconstitution, I think that probably comes down a bit to the indication and then the protein's function, and which one works best. But if you go down that route, I would say, is there an option to produce this in a single format? Like, are we close enough to where you would even consider that? And then if so, maybe the investigator, like this is all adding time at the beginning, but that's where something like Andy is saying, like, if we could do several 2 liters and evaluate this stuff side by side, that's where I think it would also be, me as a scientist says it would be interesting to look at considering an in vivo comparability between those two things, and then really making the best data-driven decision of moving forward. And then beyond that, I think if you get above, so far above 4.7, 4.8, where it's just like no chance that like no option to consider a single genome configuration, then I think that does take you down the dual vector approach, and then that's going to mean more things on the manufacturing side, like how are we going to vial it, how are we going to do potency, how are you going to deliver it. I think all of those pathways exist, but I think the first thing is really looking at like, okay, what is the size I'm dealing with, and what can I do in these two different configurations, or almost having like a decision tree around like which way to go based on initially on size, and then what does that incur with the different CMC scenarios.
Great. That covered a lot of what I was thinking about too. So maybe switching gears a little bit but also to new technologies. I mean, what about novel capsids? I mean, for groups that want to think about using novel capsids, what can be done to reduce the manufacturing risk ahead of time, before even starting to work with a CDMO? How helpful is it for groups to come in with their own like early process development data? Is it helpful at all, or is it just actually get in the way because ultimately you're just going to want to use the platform and not ignore a lot of what the client has brought to the table?
Well, I mean, I think I think having more data up front is always helpful. I think how useful it is depends probably in like what background it was evaluated, and I think this is where, like Andy mentioned, that the field is moving towards more consolidation around like 293 transient transfection based systems, and then like OEMs too, like original equipment manufacturers like Thermo and Cytiva, and like everywhere that we buy, almost all that stuff is off shelf now. 15 years ago, we were customizing everything. Like you didn't have a GMP system for doing TFF, and there weren't a lot of off shelf solutions. Now there are a lot of off shelf solutions that probably are very comparable, and a lot of people's processes are looking very similar. So I think, unless like someone comes to us and says like, hey, we did all of our research grade stuff in a back system, then like I would say, like, why did you do that? You had to make all of those vectors first of all, instead of just transfecting it. But that's where I think like the degree of that usefulness probably isn't quite as helpful. But in the case of you guys, bringing relevant data around like the harvest and affinity performance, like that's super informative for us. And I think we're still going to want to run it through feasibility, but if we can get a heads up and kind of think about where we might have some unit operation like tweaking to do, that's always helpful. So I wouldn't discourage it. I think the only thing I would say is like, don't do a lot of like pivotal dose ranging data off of that material and then have to bridge it later with the CDMO. Like just save on that, get a little bit of like manufacturability data, like maybe have an in-house mechanism or system for assessing manufacturability, just maybe for the purpose of down selection. If you're going to start with hundreds of constructs, and and then get it down to tens, like that's probably informative enough.
Where where have you guys seen engineered capsids fail? Is it that they often just fail early in production yields and or like through the platform manufacturing process, or are there risks that people need to think about that could come way down at the end when you're starting to do stability studies, things like that? Like, do you see have there been cases that you've seen where everything looks great and then you realize, oh no, it actually doesn't look great, and now we're in our GMP manufacturing?
You can't think of any time where we didn't have kind of the red flag hunting is a lot easier than like how bright is the green light. So I mean, to be honest with you, some of the hardest conversations I've had from a CDMO perspective are with really innovative, really creative capsid creators who, you know, don't want to be open to the feedback that manufacturability clearly was not a design criteria for the capsid.
Yeah.
And sometimes, like in the cochlea, truly garbage capsids from a manufacturability perspective do transformative and revolutionary work to help change lives in genetic deafness. They don't need like manufacturability is a question of need, right? Like if you only need E to the ninth or E to the 10th, that's garbage productivity. We can manage through that, but let's talk about why you know what you need. If you need to do systemic delivery in adults, oof, boy, you know. So I think we can contextualize that a little bit, but those are challenging conversations. It's one of the reasons we like the platform approach, because we could say like, hey man, we've run this 1,500 times, we've never seen results. You know, we could contextualize a little bit with data, which usually helps the contextualize the discussion.
Great.
Yeah.
Yeah. Oh, Jacob, did you want to say something?
Yeah, I was just going to briefly add, I think in my own experience too, like I think like using Andy's analogy of like the stoplight, like more often than not, like most of the unit operations are going to be pretty product agnostic. Like there's a couple that I think we know to look for, like the affinity capture step. And then if you move into like a more like our AEX based purification, like that's a for anyone that's using AEX for empty full separation, that's a pretty subtle charge based separation. We're always going to want to do a pretty significant evaluation of that step performance. I have not yet seen like an engineered capsid throw that off. And I can only think of one example, and it wasn't even at Viralgen. It was way back when I was at the university, where we had a capsid that just was brittle, like after it was held and frozen. But that's also why we would say, like, we're going to catch that early on. We're going to say, let's do in process drug substance intermediate stability as a part of your supporting studies panel going forward. Like, let's not get all the way to a vial GMP product and then figure out the capsid falls apart the first time you thaw it, or after 6 months of storage at frozen temperatures. So that's the only specifics I could think of.
Great, thanks.
If we either something real smart or real dumb, because a couple of hands are up.
Yeah, I'm just going to turn it over to Tim. Tim.
Thanks, Ben. Andy and Jacob, I co-lead this or co-coordinate this program from the NIH with PJ Brooks. And one question, or maybe it's a two-part question. If you had a suggestion for the FDA that was simple or straightforward, that would either reduce costs or reduce time in manufacturing, so lowering your burden in either of those ways, is there anything that you would suggest to them?
Kill animal studies.
All right. Okay. Anything else?
And I mean that in two ways, right? Number one, the in vivo viral safety assay itself is both expensive, harmful to animal welfare, and I've been working with it for 25 years, 20 years, and I have never seen a confirmed positive result from a viral vector product. So that one assay is also the longest tail when we look at the characterization period of viral vectors. It contributes to the delays in release of vector in a significant way, from the master cell banks to the plasmid banks all the way through, because it's done multiple times and it's also expensive. I think further agility in animal-based toxicology studies would also be, and the FDA is moving this way, right? We've seen some discussion around this, but I think in viral vector, where I don't know if it's been y'all's experience as well, but most of the heart attacks I've had in tox studies have been ultimately related to the administration of the vector or administration site issues or toxicity, in the dosing rather than the material itself. And I don't know. Sometimes I question how much more we're really going to learn, right? In additional, shooting more monkeys with a bunch of vector, sometimes I question those returns. And I see the agility that we're moving that way, but I think down in the weeds, there's additional animal study stuff to examine. Jacob, I don't know your.
Yeah. No, I mean, I'm glad you went down the route of the animal models, because I got asked that question. I did a panel a few months ago and we got asked that exact same question, and the nature of the panel was much more CMC. So I have an answer with about more CMC as well, but just to briefly comment, I think the response I gave at the time was really similar to Andy's, that in a lot of cases the animal model can be a poor predictor of human outcome. And so, I think using, and it's still, the downside here is that we're still very much this would be in development and innovative to really consider using AI-based models for in silico approaches, but even substituting the use of in vitro models in some cases, I think could be really powerful, especially when complemented with really strong and robust CMC data. Which may be a good segue into what could be standardized as far as CMC. I think the FDA and other health authorities have signaled a modernizing of regulatory science in this. At least there's been several open forums and listening forums and discussions around the idea of platforms. And, there's been guidance documents that have been released related to platform technologies as I guess as specific to the product itself, but not so much around the manufacturing technology. But, I think those of us that utilize a production platform consistently across the board with very minor tweaks, or otherwise have a well-defined platform, I think we look at that as a really clear opportunity to set bulk specifications for materials and products. And, there's huge advantages there that cuts down on the amount of supporting studies and batches and lots that need to get made early on. And to go back to the slide that Andy presented at the beginning of the call, that 1 year timeline, 10 years ago, I think people wanted to do it in a year and it was done by cutting a whole lot more corners. Now I think we're actually getting the data and the supporting documentation and CMC module that we would have taken two years to get in the past to make that a credible timeline. So I think we're actually already seeing some of that, but I think there's more opportunities even beyond phase one as we start moving into commercialization to really to leverage a platform strategy there and prior knowledge, especially when the same process and analytics have been used across the board.
Thank you very much.
Thanks for the chance to do this.
Steve, do you want to ask your question?
Yeah, can I end on a downer here? What do you do when things go wrong? Any advice if you're working with a CDMO, they make a batch, the yield isn't very good, or something happens and maybe it's a little bit ambiguous. Is it CDMO's fault? Is it just the product doesn't manufacture well? Because I mean, I have had situations like that where the CDMO comes back and says, well, it's not our fault, but I'll tell you what, if you pay for all the materials, we'll remake it for free, or something like that. If you pay for all the materials, which is most of the cost. So I don't know, any advice for kind of dealing with CDMOs in those types of difficult situations?
Um, it's interesting for me because usually those are the I spend as I've progressed in my career, I guess I've spent more time on the bad calls than the good calls sometimes. So I tend to hunt the chances like this to be on fun calls because usually like if my team is going, oh God, so I would say number one, those are the moments when CDMOs get to earn your business, right? Um, that's where you get to kind of I have never signed a CDMO contract that would not let me walk away from that situation and say, we have completed the work as contracted. Legally, we are protected. You will lose a lawsuit and say, and you're welcome, right? We as a business and any CDMO you sign, I'm telling you, every one of them, their contracts would allow that to happen unless there is gross negligence, a breach of GMP, a failure to follow the batch records. Maybe even in those cases, the CDMO will still be protected and you will probably not win the negotiation to budge on any of those terms because the reality of the CDMO business is it's asymmetrical reward for asymmetrical risk and asymmetrical reward. We are taking way less risk. We get way less reward than a drug developer, regardless of who's funding them. That's just the reality, right? So be cognizant of that, first of all, that in those situations, those ambiguous situations, you are very unlikely to be in a position to like win the argument on merit. But, and that's harsh. Like if we're going to be down, let's be down. You're not going to win the argument. The lawyers, if it gets to that point, if you're opening the contract back up, you're likely to find you're in a losing position. If anybody with any modicum of competence has been involved on the other side of the CDMO thing. Like I said, I've done it's probably over 100 MSAs, and I've never allowed those terms to budge because I can't allow my company to be endangered by a single project. I have given away a ton of free stuff though that disobeyed the contract, and I have earned business relationships that have stayed with me through multiple companies and multiple places by doing what I felt was the right thing. I think those are the moments where you get to see whether you made the right CDMO choice. And I know that's a little bit harsh, but that's where you're going to know. And a CDMO should stand behind. Now, if this was like olden times, right, where like I've had cell therapy customers, right, that swore up and down that their process didn't need development and everything was perfect and tech transfer was going to be great, right? And in cell therapy, they transfer something and it fails all the time and the customer blames you, and then you're not incentivized to collaborate because they didn't listen. In our model or in the current gene therapy manufacturing landscape, we can be held to a higher standard than that. And doing the right thing, knowing how close you guys are to patients, knowing how precious funding is, it's an uncomfortable negotiation, but that's what you should treat it as in that moment, is an uncomfortable negotiation about what the right thing to do is and how to work together on it. And if I'm being unhelpful to a future discussion that I may face with one of you down the road, I would say future commitment is the imaginary leverage you have in that scenario. Because, as I said, all I really care about is having my GMP suites full. That's where most of my people work. That's what all of us are built to do. Nobody on the CDMO side wants to fail a batch. Nobody on the CDMO side isn't trying their best, I promise. What we want is future commitment. So help us get this batch into the clinic and we'll stay with you is a really good centered spot for any customer to stay in because you're secretly giving me what I want. And that helps incentivize my ability to go do something against the contract, against my financial interests, but in the interest of like, I can sleep at night and I have a soul and I want to do the right thing. So help me do the right thing by giving me the right incentive in that situation. That's my free advice, Steve, for the worst day you're going to have working with a CDMO.
Cool. Thanks.
I don't think I have anything to add after that one. That was a good answer. I mean, things are going to go wrong periodically, though. You know, I think that maybe that is something to kind of address too, is that whether you invest in your own internal manufacturing, like I think, or you work with a CDMO partner who has um, huge amounts of capacity and works with lots of different modalities, or you're somewhere in the middle where you're like with a Viralgen or another or a similar pure player where they're only working in AAV, the risk profile changes around, you know, what that risk looks like. You know, if it's just you and you're manufacturing and you got a single suite, you could have you could have an environmental monitoring failure and have to shut your suite down because until you investigate like why the suite wasn't clean. And you know, you like that also could happen to us. We can mitigate in some cases though, and we can say like, all right, well, we've got 4 other suites and we've got an opening here. We'll move it, no issue. Unfortunately, you know, things are going to pop up where it does happen in the batch, or something that happens in another batch triggers an investigation that might have impacted your batch. And you know, I think that's the downside of being in like a multi-product facility. And again, whether it's an internal manufacturing facility or it's at a CDMO, you know, but like Andy said, no one wants to be there the day that any of that stuff happens. Um, and in fact, you know, like I've been through those investigations. It is all hands on deck. It's, you know, it is critical mass when those things pop up and people are working around the clock to try and close it as quickly as possible. Because sometimes you're in the situation where it's just stopping everything and until you can move forward. And so, um, you know, I think that touches on Andy's point of we just want to keep the suites full and keep things moving forward, because when it's not, it makes a whole lot of noise on everybody's in everybody's space. So yeah.
Well, thank you both. I know we're at time. Andy, I see you're getting ready to go. Yeah, thank you both so much for, you know, talking to us about AV manufacturing. We really appreciate all of your thoughtful answers and your transparency on many of these difficult topics. So I hope this has been educational for everyone. And I also, is it okay if we pass your email addresses around to the consortium so that they could follow up with any questions if they have them? Yeah, great.
Yeah, absolutely.
All right. Yeah, thank you both so much. This has been really fun.
Thank you.
Yeah, thank you guys
You guys.
for the opportunity.
Thanks, Ben.